Bismillahirrahmanirrahim. So, in the second part of lecture about DNA to phenotype or from genotype to phenotype, we are going to talk about the central dogma in molecular biology, how the information hidden in our DNA is decoded and eventually through the process of transcription and eventually translation, this encodes a protein. So from our hairs all the way from head to the nails of toe, we are all proteins. How a protein is synthesized depends upon how the message or the information encoded by the DNA is decoded. And this flow of information from DNA to protein is referred to as central dogma of molecular biology. So today's lecture is about decoding this information. Before we go into details of today's lecture, let's recap what we learned in the last lecture. So in the last lecture, we learned that DNA is indeed the genetic material, is responsible for heredity. We also learned about DNA replication, how DNA replicates in the S phase of the cell cycle, how DNA molecule on our chromosomes gets duplicated before cell eventually divides into two cells and passes on the equal amount of DNA or chromosomes to the daughter cells. What we learned that DNA replication follows semi-conservative mode of replication. We also talked about primase, helicase. Primase is the enzyme which adds an RNA primer. Then we learned that DNA replicates in such a way that one of the two strands of DNA gets replicated continuously while the other one discontinuously replicate, which is called lagging strand, and this is where we have Okazaki fragments. We did talk about DNA polymerase 3, DNA polymerase 1, which are involved in DNA synthesis, and DNA polymerase 1 is involved in removing RNA primer. We also talked about DNA helicase, which is responsible for unwinding the double-stranded DNA before primase adds the RNA primer. So we also talked about origin of replication, replication fork, the points on which DNA decides to start replication. And in the last part of the lecture, we talked about DNA repair, etc. Now, this is what we saw last time, that DNA is a double helix. The elongation of DNA takes place from 5 to 3' end, and the two strands of DNA are anti-parallel, so 5' and 3'. We have this sugar phosphate backbone, which is due to phosphodiester bond between one nucleotide and the next. And then on the inner side, we have these nitrogenous bases, which could be, let's say, A paired with D or G bond, making hydrogen bonds with cytosines. This is how we normally write. So if this is the DNA molecule, this is how we normally write double-stranded DNA sequence. Now, today we are going to learn how the protein synthesis takes place. This as I just mentioned, from the hairs all the way to toes, we are all proteins. One person is extremely tall, the other is dwarf. This will be topic of our next lecture when we'll start genetics. But the difference between the two individuals is a certain kind of mutation, changes in the genes which are responsible for height. But of course, otherwise, both the individuals, they have, while growing, they are making proteins. Even today in their daily lives, they are making proteins, because proteins are the molecules which perform all the functions within a cell. You can see muscular body, which means, a lot of protein there. Again, two different phenotypes. The gentleman, the sumo, must have eaten a lot of food, a lot of energy, a lot of metabolism, protein synthesis, etc. So how the information within the DNA is decoded so that cell is able to perform its function. In last lecture, we learned about that, DNA must replicate. And I gave you example of 46 chromosomes in human cell, and if a single cell divides into two cells, these 46 chromosomes in mother cell, they must be divided, replicated in such a way that the two daughter cells have identical number of chromosomes. That is only possible when each base on these 46 chromosomes is duplicated. Before this mother cell divides into two, it means there will be 92 chromosomes. Okay, and then we pass them on equally to the daughter cells. However, these cells in their normal life, they have to decode the information within the DNA, which means the code which is present in the DNA, which we know as genes, those genes need to be transcribed. So the process of decoding information from DNA into RNA is referred to as transcription. And then from RNA, so this RNA is decoded by specialized protein machines called ribosomes, and eventually we have protein. So from DNA, we have made all the way to protein, and proteins are the functional molecules, and diversity of proteins in different species are responsible for performing different functions and for the phenotype they develop. Now, what is a gene? The fundamental question you should learn from this lecture is, if we have a piece of DNA, let's say I draw it here, and this is just a very tiny part of a chromosome. Let's say we have this piece of DNA. How do we know which region of this DNA can be decoded, transcribed, which will make eventually a protein? So there are certain DNA elements which are must, which decide the structure of the gene. So on this sequence, what we have, we'll have a very specialized sequence, very specialized sequence, let's say from here to here. Okay, so I just write down 5 to 3 and 5 to 3. This very specialized sequence is called promoter sequence. It's DNA sequence A, T, C, G, T, A, T, A, etc. And this sequence is followed by then sequences and so on. So this sequence, which we refer to as promoter, is actually specialized sequence because this is recognized by very, very specialized protein, which is called polymerase, RNA polymerase, RNA polymerase. The DNA sequence here, it contains some special sequences. Let's say one of the consensus sequences TATAA, like this. Okay, we call this data box. Here, at very special position, let's say -10. Why -10? Why not -8? And from where -10 is coming? When we say this is a gene from here to here, this gene will have a promoter sequence, which I drew here, which is recognized by RNA polymerase. And this RNA polymerase then decides, after recognizing this data box and specialized sequence in the promoter, to transcribe. And so this polymerase starts sliding through the DNA. It starts moving through the DNA. As soon as it passes through the promoter sequence, there comes a specialized sequence, which we call transcription initiation site. And that sequence is +1. Anything upstream, anything above this sequence on this side will be -1, -2, -3, -4, -5, -6, -7, So data box is at -10. There are also in eukaryotic promoter, there are -35 sequences as well. They're at -35 position in eukaryotic promoter. There is consensus sequence, which is conserved in all the genes. All genes in our body will have those consensus sequence. And that's how you can define a promoter. You can tell, in computer, you can write an algorithm and you can say, okay, out of 6 billion base pairs existing in human cell, tell me where these consensus sequences are present. And these consensus sequences actually define the promoter. Wherever there will be promoter, there will be a gene. Okay, that is first thing. Then downstream of this promoter, where we will have +1, this is where when the RNA polymerase will arrive here, it will start adding first nucleotide in front of this. So let's say here is T. In here, RNA polymerase will add A. Here in the sequence, in the DNA sequence, it's C. RNA polymerase will add G, complementary. Here it is G, it is C. So transcription or decoding has started. Okay? Information hidden in the DNA is being decoded in the form of RNA. So RNA keeps moving, but one very important point you should know is RNA synthesis also takes place from 5 to 3 orientation. Now the next question is, which of the two strands, because we have 2 strands of DNA, which of the 2 strands will be decoded, against whom the complementary nucleotides will be added? So remember, if promoter is here and it's moving in this direction, so this 3 to 5 will act as template, the strand of DNA which will be copied. We call this strand the antisense strand, the one which is being copied, this one. So it's complementary will sequence RNA polymerase when it will move, as I showed you here. Let's say RNA polymerase is moving through now here through the gene body, and it will add these sequences complementary to GC, A, complementary U. So there's no T in the RNA. T, the thymine in RNA, is replaced by uridine. Okay, this one. Now, there is the strand of DNA which is 100% identical, which is not the template. We call this sense strand because messenger RNA sequence is 100% copy of this one, identical of this one. Antisense strand is the one which is copied, which is decoded, and because it's 3 to 5 and RNA has to go from 5 to 3. But which of the two strands is going to be transcribed? So let's say if the promoter sequence is going to be on this side, then which strand is going to be copied? Then this strand is going to be copied. Okay. Now, we'll talk about translation later on, but what is decoding of information? What is happening? What is role of messenger RNA? So messenger RNA is the intermediate molecule between DNA and protein. Now, once the RNA polymerase has moved all the way through the DNA, there comes a time when again within the DNA sequence, there will be some termination sequences, okay, which will be like T, A, something like a very, very specialized sequence. When RNA polymerase will come across this sequence, RNA polymerase will fall off. We call this step termination. Transcription is terminated. What was this one? This was transcription initiation phase where RNA polymerase recognized. This is transcription elongation where RNA polymerase move through the gene, and then transcription termination. Now, what you should know, what we have, how we have defined the gene structure. Gene has now promoter element, which will be recognized by RNA polymerase. Then there will be coding sequence, which will be copied in the form of messenger RNA, okay? And then there must be sequence, which is termination sequence. So what gene comprises of promoter sequences, coding sequences, which are decoded in the form of messenger RNA, and then termination sequences, which are recognized by RNA polymerase, and RNA polymerase falls off. Now, once you have the messenger RNA, this messenger RNA depends if we are in the bacterial cell. You know, in bacteria, there is no nucleus or cytoplasm. Bacteria is just one membrane-bound living organism, and then there is one big circular chromosome. Right away, there will be translation will be taking place. Now, there is a code. CUA encodes leucine. GGC encodes a glycine. CUG encodes a leucine. So each of the triplet is encoding a specific amino acid. And this is a job. Decoding from messenger RNA to this protein sequence is done by ribosomes. And eventually what we have this a protein sequence. Leucine, glycine, leucine, lysine, isoleucine. Each amino acid is linked to the next one through a peptide bond. We are going to cover both this process of transcription and translation in today's lecture. So, as I told you, your DNA on chromosome, and remember, chromosomes look like this. Each chromosome could be having thousands of genes on the DNA which it has. What is chromosome? Chromosome, I told you in the last lecture, chromosome is DNA plus histone proteins, and they are all wrapped in very, very specialized manner. They are packaged with specialized proteins called histones. The nucleosomal organization, these nucleosomal organizations of DNA, they supercoil, they go into high order structure, and eventually chromosome look like this. Now, this DNA will be somewhere here, very tiny part. This could be one gene, then another gene, then another gene, etc. Okay, many genes, hundreds and thousands of genes. Now, the nucleosomal packaging is also providing you obstacle for transcription. How are you going to transcribe? How are you going to decode? So in today's lecture, we're going to see all of this. As I told you, the RNA polymerase, the enzyme which is responsible for decoding DNA into messenger RNA, recognize specialized promoter sequence. RNA polymerase is facilitated by many other proteins, which we know as transcription factors. Okay, all of them, when this complex of RNA polymerase gets assembled on promoter sequence, then this machinery starts moving. So this is the transcription initiation phase. Now they start moving through the DNA. They come across this +1 sequence where they start adding these new complementary nucleotides. Now, which one is the antisense strand? Of course, RNA polymerase is moving into this direction. So this 3 to 5 will be the antisense, and this one will be the sense, because RNA synthesis is going to go from 5 to 3. So this one will be copied, the bottom strand. As soon as the RNA polymerase comes at +1, it starts adding complementary nucleotides. Remember, RNA polymerase is an enzyme which can synthesize RNA molecule from de novo. It does not require a primer. Like in case of DNA replication, we learned you require a primer. RNA polymerase can synthesize RNA from DNA, and it does not require primer. So it will keep going, and then it comes to a sequence where you will have the termination sequence. The RNA polymerase will hit the termination sequence, it will fall off, and it will release the polymerase. So it will release the messenger RNA sequence. Okay. So you have seen the process of transcription. What is an RNA polymerase? It's an evolutionary conserved protein complex. It's an evolutionary conserved protein machinery, whether you talk about bacteriophage, bacteria, eukaryotes, archaea, these are extremophiles. When I say evolutionary conserved, it is performing the same function. The structure of the RNA polymerase and the different proteins which come together, the subunits of RNA polymerase are pretty much conserved, although they may vary. There may be, let's say, if there are 7 subunits of RNA polymerase here, there may be 14 or 15. I may be forgetting the exact numbers, but the function of the protein is conserved, the subunits are conserved, and they are doing the same function, recognizing the promoter sequences, etc. Now, I already explained you that the RNA synthesis, just like DNA synthesis, is from 5 to 3. You have 2 strands. Once RNA polymerase recognizes promoter, it moves through. So the strand, which is 3 to 5, it will be copied from +1, which is the transcription start site, and it will start synthesizing the RNA. When the RNA polymerase will hit the transcription termination sequence, it will fall off and release the messenger RNA. Now, what is required for transcription? From the DNA, at the level of DNA, I told you we need promoter sequence. Then we need to have very specialized sequence, which we know as +1, and then transcription termination sequence. But what RNA polymerase requires to synthesize RNA? Of course, it requires nucleotides. From where these nucleotides are coming, which are going to be elongating messenger RNA? It requires RNA polymerase, and DNA we already talked about. No primer is required by RNA polymerase to synthesize. So you see the messenger RNA synthesized, that will be taken up by then another protein machinery, which is called ribosome, to synthesize protein through the process of translation. We are going to talk about this later. Let's first focus on transcription. Now, I told you the different phases of transcription. The transcription initiation, where RNA polymerase, together with transcription factors, they recognize and bind to a specialized sequence, which we know as promoter sequence, which is shown here. Okay, and promoter sequence are very, very specialized sequences, which are, if you take all the promoters in eukaryotic cell, you will find same consensus sequence, data box, -10, -35 sequences, etc. Then at initiation site, the RNA polymerase starts when it starts adding first nucleotide, and then RNA synthesis starts. That step is called transcription elongation. And when RNA polymerase comes across sequence, which we refer to as transcription termination sequence, RNA polymerase falls off, and that is called termination of transcription. So three phases: transcription initiation, transcription elongation, and termination of transcription. Okay. Now, before I move on to translation, I would like to explain you one more thing which is not physically present on the slides. There is a difference between prokaryotic gene and eukaryotic gene. Now, both are DNA sequence, both are on the chromosomes. Now, what is the difference then? We said, both have promoter sequence, both have the coding sequence, and then both have the transcription termination. So where's then the difference? Difference is when messenger RNA is decoded in prokaryotes, this coding sequence, this messenger RNA, it contains intact open reading frame. Open reading frame means when the ribosome is going to recognize, bind here, it is going to synthesize, decode, let's say, decode it into protein. Okay, right away, and it will start the, so we talked about transcription initiation site where RNA polymerase start adding sequence. Ribosome is going to add amino acid from a translation initiation site. It's a different. It will most likely be AUG sequence. It encodes a methionine sequence. From methionine, ribosome will start making protein, and it will go uninterrupted, and there will be translation termination sequence, or what we call stop codons, and we will have a complete protein. Eukaryotic gene is different. Eukaryotic gene sequences promoter, then we have coding sequence, which we call coding sequence. It is basically DNA sequence, which we call exon, intron, exon, intron, exon, intron. Okay, all this is continuous DNA sequence. A, C, G, A, G, A. After once you RNA polymerase comes across, transcription initiation sequence. Now the messenger RNA is synthesized. This is the messenger RNA. Now this messenger RNA contains exons, introns, exons. The boxes are exons, exons. So these ones are introns, exons, intron, exons. Now, it is very important to learn what the exons and introns are. Exons are basically the real, in eukaryotes, they are the real coding sequences, and introns are intervening sequences which are going to destroy your coding sequence if introns are not removed. So what in eukaryotes, after transcription, when messenger RNA is synthesized, within the nucleus, because in prokaryotes there's no nucleus, within the nucleus, okay, I try to draw it here. So this is the nucleus. Now we have a messenger RNA which contains this messenger RNA. Okay, it contains exons, introns, exon, and there is a step between after transcription and translation. That step is called, in eukaryotes, that is called RNA splicing. What is going to happen? There are specialized proteins which are going to remove these sequences. which are called introns, and they are going to join together all the exons. Exon 1, 2, 3, 4. Now, when messenger ribosome is going to bind this, so after this step, which I refer to as splicing, RNA splicing means removal of introns, and you have mature, now this is mature messenger RNA, which is only comprising of exons. This is exported into the cytoplasm in eukaryotes, and in the cytoplasm are the ribosomes, they attach and decode this into protein. Exons are the real coding sequence, which will start from a start codon and go all the way to a stop codon. We are soon going to see what start and stop codons are. So in eukaryotes, genes, the coding region of gene is interspersed with introns. So exons, introns, introns. When RNA polymerase transcribes, you have messenger RNA comprises of exons as well as introns. Introns are removed when messenger RNA is still within the nucleus in eukaryotes through the process which is called RNA splicing. And the result of splicing is mature messenger RNA, which comprises of only exons in a very specialized order, having start codon and ends with stop codon, and ribosome then decodes it into a protein. Now, what is translation? Translation, once you have mature messenger RNA in eukaryote, or in prokaryote, you don't have splicing, because it does not have, bacteria does not have introns. It's just comprised of coding sequence. And in prokaryotes, the process of transcription, in bacteria, process of this transcription and translation, we call this co-transcription translation. Co-transcription translation. As the messenger RNA is being synthesized, the ribosome catches up the messenger RNA and start decoding into protein. Because there is no splicing required here. Messenger RNA contains complete coding sequence. There's no distinction between cytoplasm and nucleus in prokaryotes. However, in eukaryotes, messenger RNA, when it's exported into the cytoplasm, now you remember the sequence of the, let's say, this is the sequence. This is the sequence of messenger RNA. And we said these sequences are to be decoded into corresponding amino acids. How do we know CUA encodes leucine, or AGA encodes lysine, or CUG encodes isoleucine? Again, through very intelligent work of people in 60s and 70s, we learned that genetic code in the DNA, what encodes leucine, what encodes glycine. So let's go now from the world of nucleotides, which are A, T, G, C, blah, 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 into amino acids. There are maximum 20 amino acids naturally existing, and all the proteins which are synthesized, they contain these 20 amino acids. And messenger RNA, which is world of nucleotides, it is to be decoded into these amino acids, which are going to synthesize proteins. Now, this is your genetic code from bacteria to humans. This is evolutionary conserved genetic code. We have 4 nucleotides, A, U, C, G. You can calculate if the genetic code comprises of 3 nucleotides. The triplet code we talked about, we said, 3 nucleotides, which is called a codon or triplet code, encodes 1 amino acids. So if you have 4 nucleotides, A, U, C, G, how many combinations you can have? You will have 64 combinations. You can have A, U, C, A, U, G, A, U, U, A, U, A. So you can just, statistically, you can calculate mathematically, and you will say we have 64 combinations. Now, what encodes those 60, what is encoded by those 64 combinations? We call the genetic code is degenerate. What is degeneracy? One thing is more than one codon encode same amino acids. For example, U, it encodes phenylalanine. C, it encodes again phenylalanine. A, it encodes leucine. G, it encodes leucine. Okay, look this one. U, it encodes leucine. C, it encodes leucine. A encodes leucine. G encodes leucine. Now, pay attention here. The first two nucleotides seem to be important, but the third one, regardless of the third one, the first two, if it's CU, you are going to encode leucine. Okay. Proline, similarly, you have proline. So in case of leucine, it's an exceptional case. We have 6 codons which can encode leucine. There is another exception, which is tryptophan, I believe. And let me see, where's tryptophan? Leucine, isoleucine, methionine, alanine, aspartic acid, glutamine, tyrosine, cysteine. Yeah, here. So tryptophan is encoded by just a single codon. Okay. But most of the time, more than one codon encodes same amino acid. So we call this degeneracy in the genetic code. And this genetic code is almost universal, as I said. Now, you can see here, these are the codons which are going to encode alanine. These are the codons which are going to encode arginine. These are the codons which are going to encode aspartic acid. And so on, you keep going. This is the leucine. This is the tryptophan, just one codon. Now, these three are very special. UAA, UAG, UGA. Here, they were listed as stop codon. Stop codon means wherever in messenger RNA they will come, ribosome will stop translating protein. That will be a signal for ribosome as a termination signal. Stop translating, stop adding more amino acid, protein is complete. Okay, wherever in messenger RNA this one comes in. Now, look at the word is coming, open reading frame. What is open reading frame? Again, let me bring you back here. In contrast to stop codon, there is a start codon as well. Methionine, only a one codon, AUG. Remember, we said transcription initiation sequence is different than translation initiation sequence. Translation initiation sequence is where ribosome will add the first amino acid, which is methionine. And in majority of the protein, the first amino acids in prokaryotes and eukaryotes, with few exceptions, is always methionine. That is like a recognition sequence for ribosome. Okay, this is from where I have to start protein synthesis. Now, what is open reading frame? Open reading frame means starting from AUG sequence, which is the first translation start codon. You go in a triplet code, you keep going, and then comes UAG, the stop codon. This can be, look, UAG can also be like this, UAG, but this will not be one codon. This U is contributing to this codon. This has to be an independent codon in the triplet code when you have such a sequence, AUG all the way to UAG in a messenger RNA sequence. We call this as an open reading frame. Now, do we only need to have messenger RNA to deduce open reading frame? No. the whole human genome and bacterial genome and all the genomes which were sequenced bioinformatically in silico, we know the potential transcription start site, potential translation start site, and what I would like you to remember in eukaryotes in particular, we have splicing sites where intron sequence is starting, where intron sequence is ending, where splicing will take place. So before this, there was an exon, the next exon. Through computer models, you can have mature messenger RNA sequence through computer models, and then tell us, okay, this is one particular gene because it contains the open reading frame. There is a promoter sequence as well. Okay. Now, when we say there are DNA is double helix, okay, reading frame, how do we calculate or how we understand the reading frame? Let's say first nucleotide in messenger RNA starts from here, CUC, AGC, triplet code. This is one reading frame. But the sequence for translation for ribosome, it may start from here, not the first nucleotide. So reading frame is not starting from the first nucleotide, but from here, the whole message is changing. Here you were leucine, serine, valine, threonine, but no more message is same messenger RNA, but if reading frame is different, you have serine, alanine, leucine, proline. You can go to next nucleotide. The reading frame may start from here, CAG, the third nucleotide, like this one. This could be the reading frame. Now, glutamine, arginine, tyrosine, histidine. So what decides from where the reading frame is going to start? As I told you, for ribosome, reading frame is going to start from AUG sequence. We will talk in the end of this lecture the mutations in the DNA. The silent mutation makes sense, nonsense, but we will talk about this later on. We will go on with the translation. Now, how many reading frames in messenger RNA can be, or in a gene model can be? If we go from on this strand, there can be 3, starting from here or from here, reading frame 1, 2, or 3. But in a double-stranded DNA, there can be 6 reading frames. How is it possible? If you have one strand of DNA going like this, and there can be 3 reading frames, there can be 3 reading frames in the opposite. So this as well. If first nucleotides, the reading frame starts, it will be reading frame 1. If on the next nucleotide, the second. So there can be 6 different possibilities. But remember, the reading frame will be in all the eukaryotes when you will have AUG. Ribosome is going to recognize the AUG, and from here, ribosome is going to calculate the triplet code. And this protein is going to, then you have the stop codon. If you say no, UGA is the first, the reading frame is starting from here. Look, you are hitting across the stop codon right away. In the middle, stop codon again. If you ignore the start codon and start the synthesis from GA, the third reading frame, you are disturbing the stop codon. You don't know where to end up. So such changes can occur if there are mutations in the DNA. If there is mutation in the DNA, you know this, let's say AUG sequence is disturbed and ribosome is not able to recognize this, you know, gene function is lost. It may be some cancer causing gene, it may be some cancer suppressing gene, etc. So in case of eukaryotes, the start codon in a reading frame is always AUG, and in the triplet code you should come across a stop codon. In case of bacteria, in addition to AUG, you have GUG and UUG as well in bacteria and in archaea. which are extreme of files. And remember, in a messenger RNA, there is only one reading frame, which contains the message, which is decoded into protein. Now, how, once the reading frame is decided, let's say this is the reading frame from here to stop codon, how the amino acids are added, how these amino acids are connected with one another, this peptide bond, and how a ribosome knows that I have to bring in methionine in front of AUG. What is connection between ribosome and messenger RNA? So there are adapter molecules, and these adapter molecules are called transfer RNA. These are non-coding RNA molecules. Okay, these adapter molecules, they read this each codon. AUG is a codon. So transfer RNA, an adapter molecule, a non-coding RNA, is going to come. tRNA is also a nucleotide sequence. It's an RNA sequence. It is going to recognize this sequence because this tRNA will have a sequence which is called anticodon. This anticodon will be UAC. This tRNA will have the sequence, and this tRNA will be also having a specific specialized site, specific sequence where methionine will be attached. We are soon going to see what is that. So this is the computer generated model, 3D model of the tRNA. tRNA is normally we call it clover leaf shaped molecule. It's a non-coding RNA. It has hydrogen bonds because the RNA molecule, the normally single-stranded RNA, can undergo hydrogen bonding within itself, and it can result in such a structure. Now, there are two specialized sequences within this tRNA, transfer RNA. This region is called, the nucleotide sequence here is the anticodon, the 3 nucleotides, which are going to be specific for, there are tRNAs for each codon. There will be sequence, anticodon, tRNA for this sequence, tRNA, an independent specific tRNA for this one. So this tRNA anticodon will recognize specific messenger RNA sequence. And on the other side, this nucleotide will be the sequence, will be the place where amino acid, specific amino, let's say this is anticodon for AUG. On this side, you will have methionine attached, for example. Okay, how it works? There is tRNA activating enzyme. We call this tRNA synthase enzyme. What this enzyme does, this enzyme, with the help of ATP, it gets activated, it gets charged. So once this enzyme is activated, it brings in an activated amino acid. Okay, this tRNA synthase enzyme. Now there will be specific specific tRNA sequence, and this tRNA sequence with specific anticodon sequence, which is specific for this amino acid, comes in on a specific catalytic site of this tRNA synthase, where it catalyzes the reaction, where corresponding to specific anticodon sequence, you will have specific amino acid attached, and then this tRNA is released from this, this charged tRNA is released, and this tRNA is ready to go and bind to messenger RNA anticodon sequence. Now, where tRNA sequence comes in? tRNA sequence, where the tRNA molecule comes in. When you have the messenger RNA sequence available for translation, protein machinery, a huge protein machinery, which comprises of many subunits of proteins called ribosomes. So ribosome has two subunits. One is called small subunit, which is this one, and the other is this one, the large subunit. First, the small subunit, it recognizes the ribosome binding site on messenger RNA. Remember, messenger RNA is what? Which contain the coding sequence. Somewhere here you will have AUG, but what are all these sequences before all these nucleotides? We call them untranslated region. These are part of exons. They are not translated. These sequences contain ribosome binding sites, recognition sites, just like RNA polymerase has promoter. This is like similar way you have sequences within the messenger RNA which provide entry site or ribosome binding site, which tells ribosome I have to translate this. And then ribosome, the small subunit, moves a bit, recognize the AUG, adds methionine, and that is the time when large subunit binds. Large subunit is divided into three different regions. For example, first region is called tRNA lending, where tRNA will come in. Then tRNA will recognize the anticodon in the tRNA will recognize its and the codon sequence. A is the amino acid carrying tRNA. Sorry, this is the landing site of tRNA, and here the tRNA recognize specific codon through its anticodon. And then as the ribosome keeps moving, ribosome is moving, you know, the tRNA comes in where the peptide bond with the pre-existing amino acid is made, and then tRNA exits from this place. In the meantime, as the ribosomes are moving, the new tRNA lands here, the other one has exit. So that's how chain elongation takes place. So this will make you more clear. What has happened now? Messenger RNA is there. Small subunit of messenger RNA binds to the ribosome recognition sequence. Small subunit moves a bit. It recognizes the first codon in the open reading frame, which is AUG. The anticodon containing tRNA, which brings in the methionine, comes in, and methionine is added. This is the time when large subunit of ribosome comes in together with the small subunit. And now what will happen? You will have new tRNA will land, recognize its codon and peptide. So new amino acid, which will recognize CCG. So GGC is the anticodon you can see. It will pair in the P site of large subunit. It will lead to peptide bond synthesis. Peptide bond between methionine and proline. And this is how then the next tRNA will come, recognize anticodon, and you will have a complete protein is synthesized like this, methionine, proline, leucine, etc., because these peptide bonds are being synthesized. Now, what will be the point where ribosome will stop translation? You remember we said there are translation termination sequence, the codons UAA, UAG, UGA. As soon as ribosome will come there are release factors, there are proteins which will come as soon as ribosome comes across the stop codon. The release factor will come in, bind to the stop codon, and ribosome will fall apart, releasing the newly synthesized protein, which is this one. Clear? Now, this is a real electron micrograph. You see many ribosomes, many. It's not just that the ribosome will catch up a messenger RNA, synthesize, then a new ribosome will come in. No. Many ribosomes, they are bound to messenger RNA, and many protein molecules are being synthesized at the same time. Ribosomes keep moving, okay? There are antibiotics, which actually blocks translation and help us against bacteria. There are different antibiotics, for example, erythromycin, it inhibits the translocation of messenger RNA along ribosome. So bacterial translation is blocked. Neomycin is also inhibiting translation. Streptomycin inhibits the initiation of translation. So that's why they say, you know, antibiotics also have side effects, because they are not just inhibiting bacterial translation, they are having effect on our cellular translation as well. Now, once translation has taken place, what is the fate of a protein? Where protein is going? Remember, each protein has specialized sequence within its amino acids, which tells the protein and the cellular machinery that, you know, let's say I have made a new protein. The amino acid sequence, there will be specialized sequence, KKRR, lysine, arginine, arginine, or specific sequence. If this sequence is present, we call this NLS, nuclear localization sequence. The cell will tell this protein that you go back to nucleus because you have to perform a function binding to chromosome or some other functions within the nucleus. There could be proteins which have to go to cell membrane and perform function as a cellular receptor. Okay, or they may have to go to mitochondria or plastids or peroxisomes. So there are specific sequences, amino acid sequence, based on which the proteins end up to destination. Normally, proteins are synthesized in the cytoplasm on rough endoplasmic reticulum, where, you know, ribosomes are attached. They bind to the messenger RNAs, and the freshly made protein is then moved to Golgi apparatus, where it is modified if it has to go to plasma membrane or released from the cell, it will be in vesicles, which will bind to cellular membrane, and through process of exocytosis, this protein can go outside the cell as well. Or this protein may act as a receptor within the cell as well. So once protein is synthesized through the process of translation, there are modifications as well. And these modifications, so now modifications of what? We are talking about modifications of protein. Let's say we have now this protein is synthesized by the mature protein is there. This protein undergoes covalent modifications. Okay, we call them post-translational modification. Post-translational, after translation, once protein is translated, there are proteins which can cleave this protein into 3 different parts or 2 different parts. This is a modification of a protein which is synthesized, and each of the cleaved part could be independently acting protein, or amino acids existing within these proteins, they may carry some sugar molecules. We call this modification glycosylation. Or let's say serine or threonine or tyrosines or histidine, whatever, they may get phosphorylation. You may have heard about lectures in cell signaling. You know, there is phosphorylation mediated communication within the cell. So all these are post-translational modifications which result in proper functional protein. For example, so I just talked about serine. So let's say there is a serine here within the protein, and this is a side chain of the serine through a protein called kinase, protein kinase. Now this serine carries phosphorylation. It carries a phosphate. Phosphorylation can be reversed as well. A phosphatase can dephosphorylate this and bring the protein back. Now it could be, this could be a functional protein, or this could be a functional protein. This depends within the cell. Yeah, here it is, for example, through phosphorylation, through this post-translational modification, this protein which is non-functional, this becomes functional. Or this could be a protein which is functional, and to inactivate, to make it non-functional, a kinase adds phosphorylation, and this protein is dormant now. It's not performing its function. Okay? So all this is part of decoding the information from DNA. From DNA, we transcribe messenger RNA, messenger RNA in eukaryotes undergo splicing. Mature messenger RNA goes to cytoplasm. Ribosomes translate. After translation, protein gets these chemical modifications, methylation, phosphorylation, acetylation on different amino acids. And these post-translational modifications may have certain specific functional consequences on protein. For example, there is enzyme. Now, these enzymes perform function. From where these enzymes come in, these enzymes are product of decoding information from DNA to RNA to protein. Enzymes are protein, with the exception of some RNA molecules, which are called ribozymes. They can be RNases as well. They can be enzymes, but with few exception, all the proteins are enzymes. No, not all the proteins. All the enzymes are proteins, with few exception. All the proteins are not enzymes. Remember this. So each enzyme has its substrate. Enzyme has catalytic site. You know, so this substrate plus enzyme, so enzyme substrate complex results in, you know, catalysis of substrate, and then, you know, this could be some metabolic product which is playing a role in photosynthesis, in mitochondria, etc. Okay. The enzymes, they do have these, you know, as I said already, the enzymes could be protein, they could be RNA. All the enzymes existing in our body, they need not to be all proteins. Some of them are RNA as well, and I mentioned already the ribosomes. The catalysis, the biological reaction in which enzymes participate, it can be enhanced if we have enzymes participating in it. So they are basically playing a role of catalyst in a biological reaction. Then each enzyme has specific substrate. Each enzyme has specific rate. You don't need to go into detail. We don't cover this in Bio 101, but what you should know is the enzyme substrate complex and the reaction of enzyme, the energy required by enzyme, and the time in which it perform catalysis, it varies from one enzyme to another enzyme, in between independent enzymes. And these Km, the rate constants, this is the rate of catalysis, how fast the catalysis is taking place, or affinity, the binding of enzyme to substrate is taking place. It's very, very specific for different enzymes, and it depends on multiple factors like temperature, pH of the buffer or the solution in which enzyme and substrate are present, salt concentration, substrate concentration. For example, if substrate is limiting, the reaction will be slow. If substrate is abundant, reaction will be fast. Amount of enzyme, if it's limiting, then again it will affect the catalysis. Then the interaction with other proteins, post-translational modification like phosphorylation. Maybe some enzyme require phosphorylation to be active. Okay, so these are different factors that affect the enzymatic activity. Some enzymes require coenzyme as well. They require cofactor as well, etc. So this is just to tell you that once you decode information after transcription and translation, that is just the beginning. Having a protein within the cell, that is just the beginning of a biological system, because these enzymes or these proteins which we have decoded from DNA, they may not be a protein. They may be a cell surface receptor. They may be a protein which is involved in, let's say, RNA polymerase is a protein. Splicing, the enzymes which are performing splicing functions, they are proteins. So the purpose of decoding information from DNA through transcription and translation is to start cellular function. And in the absence of proteins, cell is simply dead. Even if you have DNA being transcribed and translated, protein is being synthesized. If this protein is not functional, cell may die. Okay, and this is what brings us to the mutation. As I just said, you may have intact DNA, gene sequence is there, okay, messenger RNA is being synthesized, but we don't see translation there. We don't see a protein being synthesized. And this gene is, let's say, a tumor suppressor gene, very essential gene, which suppresses the cancer development in our body. Imagine DNA is intact, gene sequence is intact, it's being transcribed, it's being translated, but we don't see protein in the cell. It means there may be some mutation in the DNA. There may be some change in a critical nucleotide sequence. Maybe the AUG sequence, which encodes AUG sequence. What will encode AUG sequence? There will be T in DNA, there will be A in DNA, there will be C in DNA. What if it has become UAG? Mutation has happened. Okay. What if A is changed into U? Okay. It's no more start codon. Ribosome, when it will move through this messenger RNA, now it cannot recognize start codon. It will go to another A, because the AUG sequence can occur anywhere in the cell. As soon as it will recognize, and this AUG may not be in the same reading frame, it may be a different. Ribosome will start making protein from here, and soon it will come across a stop codon. There's no protein. Cancer development is the consequence, because this normal protein, its function was to block cancer development. So what is mutation? There are many kinds of mutation. I'm talking about point mutation. Point mutation means single nucleotide change. And a single nucleotide chain can play havoc in our lives. It may make us deaf. It may make us dead before our birth. It may lead to development of a disease later in our life. We may be living a normal life, but at certain developmental stage in our life, we develop cancer, like the breast cancer. They're living normal, all of a sudden they develop breast cancer. And the other kinds of cancers as well, diabetes, etc. Some disorders may result due to these changes in the nucleotides. And what is the consequence of mutation? For example, look at this sequence, CTA, GGC, CTG, CTA. It's 100% same sequence, this one and this one, with the exception of this mutation. This is in one individual. In other one, it has become CAT. So this was C. This C is changed to A, and it has become CAT. Now, if you look at the transcription product of this one, CUAGGC, you go like this. You have leucine, glycine, leucine, lysine, etc. What has happened? Instead of leucine, the new protein is making methionine. This is a different protein now. And due to point mutation, so this is in one individual, this is in another individual. If this leucine was a very important amino acid, this will show its consequences when this protein will perform its function. It may not be 100% efficient. It may perform only 50% of the function, or 25% of the function, or may be quickly degraded. And we will see some phenotype consequence in the form of a disease. The beauty of this example is there's protein being synthesized, there's protein being synthesized. There's no stop codon here. But this new amino acid has made this protein less efficient, non-functional maybe. Clear? So But there were other things as well. I said there could be silent mutation as well. And there could be nonsense and missense mutations. What are silent, missense, and nonsense mutations? We just talked about a mutation, change in nucleotide sequence. Okay. Now, what is a missense mutation? Missense mutation is what we learned. Let's say, where's genetic code? This one. If in messenger RNA we have a sequence GCA, if after mutation, this A has mutated to cytosine or guanine or uracil, any one of this, the third position, if adenine is converted into C, G, or U, the end result after translation will still remain alanine. So we call this silent mutation. Mutation is there, but we are lucky our protein remained same. So this mutation has no change of amino acid. So that is called silent mutation. Missense mutation is when, let's say we have this GCG, this one, we have converted into this C is converted into G. What has become GGU? Now it will not encode an alanine. It will encode something else. GGU will encode something, this one, for example. GGU is going to encode glycine. So now the sense of amino acid is changed. Now protein will have different amino acid, just like we saw. This one. We had leucine, but now after change in amino acid, methionine is there. So this is a missense mutation. And there is last one, which we said nonsense mutation. Nonsense mutation means a premature stop codon. So the third one was nonsense. Nonsense mutation means, we were having an intact reading frame going like this, but all of a sudden we have UGA. Okay. And what has happened, a ribosome started, but due to this premature stop codon, no protein is being made. And such a point mutation is called nonsense mutation. So with that, we are done with the central dogma. This is lecture. We will solve problems and some conceptual questions on Friday. So don't miss the lecture. I'm again and again saying, don't think that uploading lecture means you don't have to attend the weekly scheduled lecture, because all the conceptual problems I'm going to solve there. So this is to enhance your concept. If you don't understand anything, you can approach me anytime by my email. And as I said, these lectures are filling in the space of recitation. Okay, thank you very much.